Hello everyone, welcome back to the second part of Higher Biology Unit 2, Metabolism and Survival, and now we're looking at control of metabolic pathways. So let's consider a standard metabolic pathway catalyzed by various enzymes. Each enzyme, of course, is a protein, and the synthesis of each enzyme is controlled by a particular gene. So for example, metabolite A is converted into metabolite B by enzyme 1, which came from gene 1 and was transcribed and translated. This is the same for enzyme 2 and of course enzyme 3, which catalyzes the reaction of metabolite B to C and then on to D. However, of course, if a mutation occurs, then the metabolic pathway might well be disrupted. Gene 1's okay, its DNA is intact, no mutations there, so enzyme 1 is produced, so metabolite A is catalyzed to metabolite B. However, let's say gene 2, which is barely visible, a mutation occurs, it's incorrectly transcribed and translated, and of course the enzyme produced is non-functioning. So the metabolite B, the levels of metabolite B within the cell might well build up and will cause whatever problems it may. If we have a brief look back at genetic control, of course the role of genes is to make proteins. All proteins help to form a cell's basic structure, or for enzymes to control metabolic processes, or lots of other different types of protein, as we have seen. Okay, so each gene is coding for a different protein. Now genes can be switched on or switched off depending on whether the protein that those genes code for is required. Okay, so there's the genes being switched on and switched off. Now a great example of genes being switched on and off, as it were, is to do with the action of a beta-galactosidase enzyme on lactose sugar. If we look at lactose to begin with, it's a disaccharide made of glucose and another sugar called galactose. And of course the beta-galactosidase is the enzyme that breaks down lactose into glucose and galactose, breaking that bond in the middle like so. So essentially beta-galactosidase is lactase. Now the scientists who came up with this model of lactose metabolism, scientists called Jabob-Monod, and their hypothesis, so a bit of background information for it anyway. Glucose, of course, is the primary respiratory substrate for organisms, and the organism they were looking at in their model was Escherichia coli. Now E. coli can only use the glucose in the lactose if it's first broken down, if the lactose is first digested. But the E. coli only produces the beta-galactosidase if lactose is present. So the presence of lactose switches the gene on for the production of beta-galactosidase, and this is known as enzyme induction. The production of the enzyme has been induced by the presence of the substrate. Now, an important aspect before we look at how that occurs is this, what's called an operon. On the DNA, the part of the DNA that's associated with the production of the beta-galactosidase enzyme, an operon itself contains structural genes, and also next to it an operator gene which controls the structural gene. There is also, related to this, a regulator gene which produces what's called a repressor molecule, and that plays its part in the induction of the beta-galactosidase enzyme, as you will now see. So here's the DNA chain associated with the enzyme. There's the actual gene that codes for the structure, the actual enzyme itself, and there's the operator gene right next to it. Somewhere else along the DNA strand is the regulator gene, and that produces the repressor molecule. Okay, so let's have a look at how this works and how the enzyme is only produced when lactose is present. So the regulator gene is transcribed and translated, and the repressor protein is produced. Now, normally, no lactose is present. That repressor molecule will combine with the operator, or latch onto the operator, and it will prevent the structural gene for the beta-galactosidase from being switched on, as it were, so the enzyme is not produced. However, what happens is, if lactose is present, there's the lactose molecule suddenly appeared. This repressor molecule now binds to the lactose, so the operator is free, and the beta-galactosidase is produced, and the enzyme is formed. But once all the lactose has been digested, then that repressor molecule once again binds to the operator, because there's no lactose left for it to bind to, and that structural gene for the production of the enzyme is switched off. Okay, now that process is often referred to as the lack operon, the lack for lactose. Now, some metabolic pathways operate continuously, and the enzymes that catalyze those reactions, the genes for those, are constantly switched on, and they're always present. Now, these metabolic pathways, regulation of their enzyme action can be brought about as follows. Firstly, defective signal molecules. For example, adrenaline produced by the adrenal gland it basically initiates the signal molecule to initiate the breakdown of glycogen into glucose in the liver cells, and that glucose is then put out into the bloodstream, like so. So let's have a look at that. There's the adrenal gland, there's the red blood cells flowing through a blood vessel. Some stimulus to initiate the fight or flight response means that the adrenal gland produces adrenaline. The adrenaline moves into the blood, attaches to receptor proteins on the surface of the cells in the liver, and this initiates the breakdown of glycogen into glucose, and then the glucose produced moves into the bloodstream and is carried around to all your body cells. If we now have a look at the effect of inhibitors, these decrease the rate of enzyme-controlled reactions. There are three different types: competitive, non-competitive, and feedback end product inhibitors. Now, if we look at competitive inhibitors to begin with, these compete with the substrate itself for the active sites of the enzyme. So if you increase the concentration of the competitive inhibitor, that will reduce the rate of the enzyme reaction. For example, there's a standard rate of enzyme reaction without an inhibitor and with an inhibitor present. In order to overcome the effects of the competitive inhibitor, you have to increase the substrate concentration further. Non-competitive inhibitors, they work in a different way. They don't compete with the substrate for the active site. They bind to another part of the enzyme called the allosteric site. And in this way, non-competitive inhibitors can act as regulators. So here's your inhibitor, binds to the allosteric site, and that alters the shape of the active sites of the enzyme, so it no longer is complementary with the substrate. So now it can help to control the rate of enzyme reactions. Finally, I will look at feedback inhibition by a particular end product. So another metabolic pathway here. Metabolite W is catalyzed by several enzymes up to metabolite Z. And once metabolite Z reaches a certain level, in order to prevent further production of metabolite Z, it acts as an inhibitor for enzyme 1. So it stops the metabolic pathway at that point. Okay, thank you very much indeed once more.